So now I'm gonna hand it over to Aden. No lab, no code chain, no bioinformatics, real-time eDNA species identification from a backpack.
So hi everyone, happy to kick things off as a first speaker. I'm Aden Ip. I am a research scientist and a principal investigator at the eDNA Collaborative at the University of Washington. So my talk today it's mainly about environmental DNA and conservation applications in the marine world. First off, environmental DNA. I'm quite certain everyone at least have heard of this term before, but very briefly, every organism sheds DNA into the environment, into the surroundings, into the soil, water, and even the air. And if you can collect it, you can identify what species are present without ever seeing or capturing the animal. And to me, I think this is an incredibly powerful conservation application too. And the barriers to entry have always been high. So theoretically, whenever you want to do eDNA work, you need like a fully, functioning lab, cold chain storage, and a kick-ass bioinformatician. So right now we're trying to navigate around this, high barriers. We're trying to lower the barriers of entry to build a system that reduce the reliance on these things. This is what a typical eDNA workflow looks like for many people today. First you collect an environmental sample. Most of the time it's a water sample. You freeze it and then you ship it to a lab. Somebody else extracts DNA. Somebody does a PCR and then sequence on, a fancy machine. And then, a coder, a bioinformatician runs it through computational pipelines that's like a black box. You don't really know what's going on. And then weeks to months later, you get a species list back. And by then you have left your field site, follow up, immediate follow up is not possible. You also can't adapt your sampling in real time. And sometimes the information might arrive too late to be, readily actionable. And for most eDNA teams, I think the bioinformatics alone is a deal breaker because you really need someone who can code, who can assemble a local database that's relevant to the area that you are trying to survey, and also who can troubleshoot a pipeline and talk to the computer. And most field teams don't have the person. So my team right now, we ask this question: what if we remove all of this, restrictions and simplify everything? Can we still get it to work? And we have actually designed a fully portable prototype that fits in a single backpack with no cold chain and also automated bioinformatics expertise. So for the moment you collect a water or air sample, you can see species names appearing on the screen with a turnaround time of about 4 to 6 hours. This is a recent breakthrough from my team, and I think that is the fastest eDNA meta barcoding results turnaround at a community level reported anywhere. We have also designed improvements like in the wet lab, like you fill the water sample, you extract, amplify the DNA, and then you simplify protocols with less reliance on cold chain and load it on the nanopore sequencer, which is here over on the right, is the size of a stapler. And yeah, everything runs on batteries and solar power. So all you need to do is to look for a sunny place, and then your species names will appear as your environmental sample is being sequenced. And we have been trying to stress test this over several deployments. We deploy it at extremes of the infrastructure spectrum. On the left, we have started with DNA barcoding on a coral reef research cruise, and then we moved on recently this year to do this whole workflow across the Western Pacific from Philippines to Guam on the research cruise. We did real-time eDNA barcoding, and we detected vertebrates, invertebrates, even taxa associated with deep sea hydrothermal vents, all processed at sea. And then on the right, we also ran this workflow in the Amazon rainforest. We picked up Plasmodium, which is the malaria parasite from canopy eDNA swabs. And also with the same workflow, same backpack. In fact, starting today, I'm going to try to deploy this on an offshore island in Washington State in the United States with no power, no lab, and no connectivity. And then we'll do water and air eDNA process entirely off-grid on solar power and batteries. So that being said, we're also trying to remove the last barrier, which is, sometimes we don't even need to sample water at all. And because sometimes there's really no water to sample, or it's just too dangerous to sample the water, or, yeah, it's just impractical. But at least the DNA is still in the environment. And most of the time it's also in the air, it's recoverable from the air. So water consistently, throws DNA upwards, there's splashes, there's ripples, there's bubble bursting, and that aerosolizes waterborne DNA into the air. Terrestrial species also shed DNA in the air directly. So air eDNA can capture both aquatic and terrestrial biodiversity in a single sample. And we have published a study. We did this test, proof of concept study over a salmon spawning creek with passive air eDNA samplers that require no power, no pumps, and cost less than $10 each. We hang up, come back the next day. And from the same paired air and water sample, we found 35 vertebrate species across air and water. 40% showed up in both media. And you see like even fish in the air and also mammals in the water. It's pretty cool. And also the airborne signals are tracked with actual fish counts week by week. So now I just wanted you to picture if you pair that with a backpack system, and like $10 sampling tray, you can do field sequencing and gives you species names in hours. And that's really amazing to me. And you can actually establish a monitoring network that, they can put in places that are too remote or too dangerous or too expensive to reach in any other way. So with that said, we built this system, which is great. It's automated, it's off-grid, but I realized that I can't scale it alone. I'm actually actively looking for field partners. If you're interested to apply eDNA, you manage a protected area, a coastline, or anywhere you need biodiversity data that a lab can't get to, I'm interested in collaborating to bring this backpack to your site and run it with your team as a real deployment, customized, to answer your questions, detect your species, and look at your data. I'm also looking for engineers because this system works in the Pacific Northwest. The biology is working, but I have not looked into thermal management, into tropical heat, weatherproofing for monsoon seasons, and all the shenanigans. Also beyond species detection, we're also moving on to try to resolve individual animals from seawater. That's a longer conversation, but I'm happy to take this offline if anybody wants to know if you can do eDNA and population genetics. I have to jump off a field after this, but if anyone has more questions and want to collaborate more, just feel free to drop me an email or scan a QR code and you get my email. Thank you.
Thank you so much, Aden. We have a few minutes for questions. I love this presentation because we recently published Talia will mention about that later, but our State of Conservation Technology survey findings suggest that many practitioners are really excited about eDNA, but still find it difficult to implement in practice. So I think you have shown how you remove many barriers, and I wanted to ask you, what do you think is the biggest thing holding back wider adoption today? And if this has changed now that technologies like your portable sequencing is becoming available?
I think there are many, it's a like multifaceted problem, but I think the very first thing is sampling. Like people don't know how to sample eDNA. And also, like the frequency, how many samples you need um, how many samples do you need to take to detect the target species. So that actually requires collaboration with relatively experienced people with eDNA. And also, yeah, I think that's the main problem. And then bioinformatics also spooks people out. So I think that's the two main things that are the main barriers, because the wet lab part can actually be outsourced to commercial companies, but that will be a time lag of like 2 to 4 weeks. So portable sequencing is great because it's going to be 4 to 6 hours, like I said. Yeah.
Thank you, Aiden. I think we can. Matt, do you want to ask your question? We can make time for one more question.
Yeah. Aiden, two-part questions, Matt Pickett. Is this commercially available or is something that you're creating, in the lab and there's one of one? And I missed what you said about saltwater. We have a upcoming collaboration down in St. Martin to do some eDNA work. And right now the plan is just to send it to the lab. But wanted to get your thoughts on that.
Yeah, it works with any workflow, air, freshwater, saltwater. We just have not done anything with sediment. It's not commercially available yet. There are plans to make it commercially available. We just finished um, assembling a prototype like 2 weeks ago, and we are doing like our final round of stress testing literally today. So I'll keep you in the loop. We can, keep in touch over email when, I have more progress on getting it up down the commercial chain.
Thanks, Aden.